Welcome to AP Biology. This video is on the stages of cell respiration. Cell respiration is going to take place in three major stages. Stage number 1 is called glycolysis. Stage 2 is pyruvate oxidation and the citric acid cycle. Stage 3 is called oxidative phosphorylation. That includes electron transport and chemiosmosis. Here we see an overview of what's happening during the process of cellular respiration. This is all taking place in the presence of oxygen. The first step of cell respiration is called glycolysis. You can see here that during glycolysis, glucose is going to be converted into pyruvate. This happens in the cytosol of the cell. It does not take place inside the mitochondria, and glycolysis is going to happen regardless of whether or not oxygen is present. Glycolysis makes a small amount of ATP, small amount. In the presence of oxygen, O2, pyruvate is going to enter the mitochondria. And the first step it's going to go through is called pyruvate oxidation, where pyruvate is converted into acetyl-CoA. Acetyl-CoA will then enter the citric acid cycle, also called the Krebs cycle. The citric acid cycle also makes a small amount of ATP. The goal of these three stages, these three steps here, are not to make ATP. The goal of these three steps are to oxidize glucose, to remove the electrons and transfer those electrons to NAD+ to make NADH. Remember, NADH is an important electron carrier. It's carrying the electrons that came out of glucose. The goal of these three steps is to build up the amount of electron carriers, the amount of NADH. All of those NADHs are going to carry the electrons over to a structure called the electron transport chain. The electron transport chain is where oxidative phosphorylation takes place. Oxidative phosphorylation is where a bulk of the ATP is made during cell respiration. There are two processes by which ATP is made. One's called oxidative phosphorylation. We'll see this in more depth in a little bit, but it's using oxidation, the removal of electrons, in order to make ATP. Or in other words, in order to phosphorylate ADP to regenerate ATP. So oxidative referring to the oxidation of glucose, and phosphorylation referring to the regeneration of ATP. A small amount of ATP is made through a process called substrate level phosphorylation. This is going to happen during glycolysis. This also happens during the Krebs cycle. During substrate level phosphorylation, an enzyme is used to manually add a phosphate onto ADP to make ATP. This phosphate is coming from a substrate in the chemical reaction. So it's a physical process of regenerating the ATP. And again, only a small amount of ATP is made through this substrate level phosphorylation. We begin with the process of glycolysis. Glycolysis is going to happen in the cytosol. Glycolysis is going to happen with or without oxygen. During glycolysis, glucose is going to be split in half, and it's going to make 2 molecules of pyruvate. Glucose is a 6-carbon molecule. It gets cut in half to make 2 3-carbon molecules. Each of these 3-carbon molecules we call pyruvate. Every time a chemical bond is broken, what we are performing is the process of oxidation. We are removing the electrons. Those electrons are going to go to NAD+ to form NADH. So glycolysis is the first step in which glucose becomes oxidized. Glycolysis has two major phases, an energy investment phase and an energy payoff phase. The first half of glycolysis is the investment. 2 ATP get invested to split the glucose in half. During the energy payoff phase, 4 ATP are made. So if we look at glycolysis overall, it has a net gain of 2 ATP, and those 2 ATP were made during substrate level phosphorylation. Since we're breaking a bond in glucose, we are oxidizing it, we are removing the electrons. Those electrons are going to go to NAD+. NAD+ gets reduced to make NADH. So glycolysis is going to make 2 ATP, it's going to make 2 NADH, and it's going to make 2 pyruvate, 2 3-carbon molecules. In the presence of oxygen, so we're going into aerobic cellular respiration, that pyruvate will now enter the mitochondria. If we look at the structure of the mitochondria, here we see the outer membrane, here we see the inner membrane, and in the middle, the central portion of the mitochondria is called the matrix. In the presence of oxygen, pyruvate will enter the mitochondria and it will go directly into the mitochondrial matrix. This is where pyruvate oxidation takes place. Pyruvate is going to get oxidized. A chemical bond is going to be broken. When that bond is broken, those electrons are going to go to NAD+ and form NADH. When pyruvate gets oxidized and the electrons go to NAD+ to become NADH, the resulting molecule is going to be called acetyl-CoA. So we're going from a 3-carbon molecule to a 2-carbon molecule. That third carbon is going to be released as carbon dioxide. Carbon dioxide is a waste product of cell respiration. It can't be used for energy because it's been completely oxidized already. In order for pyruvate oxidation to take place, we need the aid of a coenzyme called coenzyme A. So overall, 2 pyruvate are going to enter, making 2 acetyl-CoA. 2 NADH are made, 2 carbon dioxide are made. Acetyl-CoA will then enter the citric acid cycle. The citric acid cycle, or the Krebs cycle, is where glucose becomes fully oxidized. The final bonds are broken. Those electrons are all going to be transferred over to NAD+ to become NADH. Let's look at the citric acid cycle. So here you see 1 acetyl-CoA entering. 2 are going to enter total, so we could double all these numbers. You can see that each acetyl-CoA has 2 carbons. 2 carbon dioxides are released. So altogether you're going to have 4 carbons enter, 4 carbon dioxides are produced. Therefore, that glucose is completely oxidized. There's no more carbons left. There's no more molecule. It's all been released as this carbon dioxide waste product. As a result, 6 NADHs are going to be made total. We also have the creation of another electron carrier called FADH2. One FADH2 is made per acetyl-CoA, so you're going to have 2 made total. You're also going to see 2 ATP made, and the ATP made during the Krebs cycle is also made through substrate level phosphorylation. So at the end of the Krebs cycle, we no longer have any more glucose. All those carbons have been released as carbon dioxide. All the electrons that have been ripped out of glucose systematically are all being carried by NADH and FADH2. They are going to carry those electrons over to the electron transport chain. The electron transport chain is going to be where oxidative phosphorylation takes place, the process that's going to produce the majority of ATP during cell respiration. The electron transport chain is located in the inner membrane of the mitochondria. The inner membrane has many bumps and grooves that increase the surface area, allowing the mitochondria to have more electron transport chains, and each of these bumps and grooves is referred to as the cristae. The electron transport chain is physically made up of a group of proteins. These proteins are called cytochromes. As the electrons go down the electron transport chain, they are eventually going to end up with oxygen. We know that oxygen is going to gain the electrons, it's going to gain the hydrogen, and it's eventually going to become water, the final waste product of aerobic cellular respiration. Here we see a picture of the electron transport chain. So if you're looking at this picture here, this light yellow region down here, this would be the mitochondrial matrix that's in the center of the mitochondria. Here you see the inner membrane space. That's the space between the inner membrane and the outer membrane. Here we see the inner membrane. You could also call this the cristae. To show you it better, we can go back to our picture of the mitochondria. So again, here you have the outer membrane. Here you have the inner membrane. Here you see what a cristae looks like. In between the two we have the inner membrane space, and in the center here we have the matrix. So what we're looking at, the electron transport chain, is actually located right in the inner membrane, right here. NADH and FADH2 are going to carry the electrons to the electron transport chain. At the electron transport chain, NADH gets oxidized. It loses its electron. It's also going to lose its hydrogen ion or proton. The electron is then going to travel down the electron transport chain. And as it moves from protein to protein or cytochrome to cytochrome, that electron releases energy. Some of the proteins in the electron transport chain are proton pumps. They are proteins that pump protons using the power released from that electron. All the protons that have been released from NADH as it got oxidized are all pumped into the intermembrane space. This is a good example of active transport, and this creates a proton gradient, just like putting water behind a dam. This proton or hydrogen ion gradient that you see right here we refer to as a proton motive force. It's going to have the ability to perform some work, and we'll see that work in a minute. What's moving the electron down the electron transport chain? It's oxygen and oxygen's electronegativity. Remember, oxygen has a strong pull of electrons. Also located in the inner membrane is an important enzyme called ATP synthase that you see right here. ATP synthase is a protein channel that has a turbine attached to it. When the protons create this gradient, this proton motive force, the ATP synthase is going to open up, and all these protons are going to rush through the ATP synthase. When they rush through the ATP synthase, they are going to spin the turbine like you see right here. Spinning that turbine is going to be used to add the phosphate onto ADP to make ATP. This process in which protons move from high concentration to low concentration through the ATP synthase is called chemiosmosis. Chemiosmosis is a really important key term in this unit because it is the method by which ATP is directly made. It's made through the process of chemiosmosis. The protons diffusing down their gradient, spinning the turbine of the ATP synthase, making the ATP. Going back to our diagram here, you can see that the protons going through the ATP synthase has been used to manufacture the ATP. After the protons have gone through, the protons plus the electrons will go to oxygen, and oxygen will become water. Oxygen is referred to as the final electron acceptor. All those electrons began with glucose and they are all ending with oxygen. Oxygen is getting reduced and it is becoming water. So here you see two processes taking place. One is going to be the electron transport chain. The electron transport chain is used to make the proton gradient. Then that proton gradient is then used to make the ATP. Those protons diffusing down their gradient is called chemiosmosis. This entire process, combining the electron transport chain and chemiosmosis, is what we call oxidative phosphorylation, because ultimately we're using the oxidation of NADH, and really we're using the oxidation of glucose in order to manufacture ATP. This is going to be how a bulk of the ATP is made during aerobic cell respiration. Here we can look at where all the ATP is made during aerobic cellular respiration. Very little is made through substrate level phosphorylation. You have two made during glycolysis, you have two made during the citric acid cycle. Oxidative phosphorylation that takes place combining the electron transport chain plus chemiosmosis makes 32 ATP per glucose. So you can see this is a bulk of the ATP production. To look at cell respiration altogether, all these steps that we just went over are systematically removing the electrons away from glucose. You are oxidizing glucose. Every time glucose gets oxidized, those electrons are transferred over to NAD+ to make NADH. NADH is an electron carrier. It's going to carry the electrons over to the electron transport chain. At the electron transport chain, NADH gets oxidized. It removes its electrons, it removes its protons. As those electrons go down the electron transport chain, the energy released is going to be used to make the proton gradient, or our proton motive force. Those hydrogen ions, or those protons, then diffuse down their gradient through the ATP synthase through a process that we call chemiosmosis. And chemiosmosis is going to make the ATP. All of those electrons that came from glucose are going to end up with oxygen. So ultimately you have the oxidation of glucose, the reduction of oxygen. Oxygen gets reduced, it gains the electrons, it gains the protons, and becomes water. So if we look at the three major products of aerobic cell respiration, you have carbon dioxide made as glucose is oxidized, you have water, and you have energy. In this case, we have a combination of ATP and heat that are produced. This concludes our video.